Look at the screen right now. You are reading words and recognizing shapes. Think about which part of your body is doing that processing. The standard assumption is that your eye acts a lot like a simple digital camera. It supposedly captures raw pixels and passes them back via a cable to your brain, where the real computing happens. But the biological reality is completely different. The retina at the back of your eye isn't an isolated sensor. It is living neural tissue. Anatomically, it is a direct extension of your central nervous system. This means that some of the most intensive calculations required to make sense of the visual world begin happening right inside your eyeball, long before the data travels anywhere else. The sharpest part of that processing takes place in a tiny region near the center of the retina, called the fovea, which measures roughly 1 millimeter across.
Because incoming light is focused directly onto this specific spot, the fovea handles our most demanding tasks. It is the tissue that allows you to read a book, thread a needle, and recognize human faces. This 3D representation shows that 1 millimeter foveal pit, zooming down into a microscopic sample block near its center. Scientists have now successfully mapped the exact nanoscale wiring inside the specific block of tissue, creating a comprehensive structural map known as a connectome. By tracing these microscopic pathways, researchers can finally see exactly how the eye filters a chaotic visual environment into a clean, organized signal before the brain ever gets involved. Capturing biological structures this small requires moving past standard optical microscopes, which simply cannot resolve details at the nanoscale. This animation illustrates the physical scale of the experiment. The team took a microscopic cube of human retinal tissue, just 180 by 180 micrometers wide, and physically sliced it into over 3,000 individual sheets, each only about 50 nanometers thick. They imaged those sheets at a 5 nanometer resolution. Then, relying heavily on artificial intelligence and computational algorithms, they analyzed the images to predict exactly where the microscopic borders of one cell end and another begins. When the slices were reassembled into a 3-dimensional volume, the data revealed approximately 3,000 distinct reconstructed cells packed tightly into that isolated block. Even more remarkably, by scanning those cellular branches, researchers identified over 300,000 individual synapses bridging those cells together. Cataloging cell types gives scientists a biological parts list, but mapping the exact location of hundreds of thousands of synapses reveals how data actually flows through a living network. You might expect the architecture for high-resolution human vision to look like a massively parallel maze of wires, carrying every possible piece of data simultaneously. But looking at this diagram of the reconstructed circuitry, a surprising simplicity emerges. Researchers identified 11 different visual pathways at the retinal ganglion cell level, yet an overwhelming 95% of the foveal output from cone-driven excitatory circuits funnels into just 3 major routes. This architectural bottleneck demonstrates that the retina isn't preserving every raw pixel it captures. It actively sorts, organizes, and compresses the visual data. Instead of a sprawling, unorganized grid, the fovea operates like an ultra-efficient information expressway. This dense structural map also challenges long-held assumptions about how human color vision operates. This model illustrates a highly specific anatomical finding. The researchers discovered evidence for physical, electrical couplings connecting short wavelength S cones directly to adjacent medium and long wavelength cones. According to the researchers' modeling, this implies that signals from different cone types can interact and blend before the very first chemical synapse in the visual pathway is even triggered. The biological foundation of our ability to perceive vibrant colors is being computed at the absolute first anatomical step of sight. We do need to establish a reality check here. This study represents a draft connectome. It maps a tiny sample of tissue, not every neuron in an entire human eye, and the data is still undergoing proofreading by the scientific community. So why invest years of scientific labor and extreme computational power into mapping a microscopic speck of tissue if it doesn't decode the entire visual system? This split-screen comparison shows exactly why it matters. Medical science cannot fix diseases that cause central blindness if doctors do not know what the healthy architecture is supposed to look like in the first place. This draft connectome provides a critical biological reference point. It gives medical researchers a precise, physical blueprint of the machinery, highlighting exactly where future interventions and treatments need to act. Let's pull back from the static map and look at the living system. Every single time you look at an object, light enters your eye, and hundreds of thousands of microscopic synapses shape that incoming signal in a fraction of a millisecond. If this much intense physical calculation is happening entirely inside the retina, how much of what we call seeing is completely finished before the brain even knows it's happening? Let us know in the comments what part of the human body you think science should map next. If you want to keep exploring the incredible machinery inside you, make sure to like this video, subscribe to the channel, and we'll see you next time.